What is associative learning? Associative learning is a type of learning about the relationship between two separate stimulus. These stimulus may range from tangible objects to abstract concepts. Associative learning theories strive to capture the processive underlying and driving the changes in strength of the association between representations of stimulus that develop as a result of experience of the predictive relationship between those stimulus. Historically, formal models of associative learning have focused on two potential factors underlying associative changes, namely processing of the conditioned stimulus in terms of changes in associability and processing of the unconditioned stimulus in terms of changes in error. This concept of classical conditioning in association with smoking-related topics serves as a way through which the development, maintenance, and relevance of general addictions can be explored, so that innovative solutions and experiments can be designed to address these issues. Several studies have found that initially natural stimulus directly associated with smoking can bring out conditioned responses. However, few studies have been conducted to demonstrate the contribution of higher-order conditioning direct learning in the outside world.
In an empirical study, Little and Franken investigate the relevance of associative learning in regard to addictive disorders, specifically exploring higher-order learning practices associated with smoking dependency and its electrophysiological correlation. Motivated by recent discoveries made by Franken and peers, the objective of Little and Franken is to evaluate higher-order classical conditioning procedures related to smoking dependency by using straight cortical measures, namely brain action as assessed by ERPs. Franken and peers critically identified that conditioning practices can be measured with ERPs, which are electrophysiological brain responses, to internal and external stimuli, particularly suited to study differences in cognitive processing and thus providing information about the extent of neural engagement, hence framing the research approach adopted by Little and Franken.
The research method was characterized by 61 participants, among whom 30 were smokers and 31 were non-smokers. The criteria for classification rendered individuals who smoked less than 5 cigarettes within lifespan to be considered as non-smokers, while those who smoked an average of 10 cigarettes a day to be characterized as smokers. Experimental trials consisted of 40 smoking-related pictures containing cigarettes and other smoking-related objects, and also another 40 neutral pictures lacking any connotation to smoking. These pictures were paired alongside two geometric objects, a green pyramid and a red cube, which appeared first before either a smoking or a non-smoking related picture emerged onto the screen. Thus, a conditional stimulus is formed where a connection between smoking and non-smoking stimuli with the two respective geometric figures is unconsciously established for the participants. During experimental procedure, participants were required to detest from smoking to prevent impact of nicotine onto ERPs. Once trials were completed, each participant was sent to a light and sound attuned room where they were instructed to sit still and carefully observe presented stimuli without employing distracting thoughts, all while electrodes measured any occurrence of neural stimulation, such as any signals of arousal or response.
Upon completion and analysis of data collected, results reveal that individuals classified as smokers display a recurring trend of processing bias towards stimuli associated with smoking. Additionally, researchers also recognized that neutral figures would also evoke equal conditioned responses, demonstrating occurrence of second-order associative learning. Furthermore, analysis from ERPs confirmed that incidence of second-order associative learning was increasingly prominent in individuals who were categorized as smokers, with obvious displays of greater approach bias, attention bias, and psychological reactions.
Overall, Overall, the conclusions from the study are highly limited. They do provide insight into the occurrence of high-order conditioning in the context of smokers. Although these findings are introductory, they allow for greater understanding of smoking addiction and generate potential for future research and application of addictive disorder research.
Although Liedl and Franken provide an effective representation of the relationship between addiction and associative learning, they also highlight respective weaknesses and strengths within the study that influence the depth and real-world applicability of the study itself. Structurally, maximal consistency is maintained through the classifications of control and testing groups of similar demographics and through the use of combined empirical measures. By ensuring that there are 30 participants in each category with a mean age of 20.5 years, are selected from the same Dutch university and classed with similar smoking rates, investigators avoid the onset of age and experience-related obstacles that may confound natural results. Alongside this, the employment of computerized self-report and analysis software measures prevent the genesis of human error and measurement and investigator bias. These precautions serve to preserve the accuracy, validity, and reliability of the study and in turn enhance the legitimacy of its wider world application. A signature strength is the extensive analysis of past research conducted by Liedl and Franken. This analysis of gaps prompted the development of an associative stimulus experiment with the inclusion of a neutral stimulus which had not been previously done. Innovative design introduced new paradigms that considerably expanded the current sphere of psychological research. As a subsequence of the findings on electrophysiological correlations between higher-order conditioning and nicotine addiction, hints as to the connection between which stimuli encourage and maintain subconscious compulsions across addict and non-addict brains are developed. On a clinical level, this broadens the development of new paradigms that further explain the neurophysiological and psychological basis of behaviors that contribute to the enhancement and expansion of the current field and future fields of research and analysis, while on a more applicable level can aid the growth of new campaigns and prevention strategies by governments and medical bodies that aim to address particular health concerns such as drug-related and generalized addiction morbidities and mortalities. However, these developments are undermined by the study's limitations. A key weakness involves the study instructing individuals to abstain from smoking for a minimum of an hour before the experiment as a way of avoiding nicotine contamination of ERP signals. In just 20 minutes of cigarette abstinence, blood pressure and heart rate stabilize and a 35% increase in oxygen presents, which enhances EEG changes. These physiological changes work alongside psychological shifts such as increased irritability and difficulty concentrating to moderate results as individuals are subjected to perform under anomalous circumstances that do not reflect their typical processing capacities, hence providing inaccurate results. Further, by explicitly telling subjects to formulate stimulus associations, both placebo and effort and efficacy effects are encouraged, wherein due to this conscious awareness, individuals are motivated to formulate connections that aren't naturally present, which produces falsified results that on a preliminary level are deemed unreliable and inapplicable. Furthermore, the lack of inclusivity regarding age differences and differences in the severity of nicotine and addiction and consistency across gender distribution as a subsequence of the college sophomore paradigm. The reliability and functionality of the study's results are inhibited as unaccounted for moderator variables prevent the study from making broader contributions regarding the topic of association and addiction, as differences in performance that stem from these factors are not actively present, which fails to be reflective of the wider populace and individual contexts that produce divergent results in forms of this study.
We propose a new study that accounts for some of the aforementioned weaknesses, aiming to offer results that can give more direction to smoking addiction management by further understanding relevant associations. More specifically, the aim of the study is to lay a groundwork of information regarding specific demographics of people. Demographics based on factors of age, gender, severity of addiction, and duration of addiction. As earlier mentioned, the previous study had minimal diversity in participants and lacked the depth of having moderator variables. The new study will acquire participants over a much larger demographic range, having moderator variables of age and gender. This will serve to identify the differences between demographics on a larger scale. The benefits of the study mainly focus on its potential to obtain much more meaningful results. This is that the results could be used to formulate techniques of addiction management that can be specialized for and thus provide further efficacy towards treatment of certain groups of people. Furthermore, this could provide preliminary data for future studies towards understanding how addiction functions beyond areas than just smoking. Participants would be chosen at random and receive a questionnaire to ensure they are eligible for the study. This questionnaire would also determine the values of each participant's moderator values, including information about how much they smoke and how long they have been smoking. The experiment would last a month, with each participant filling out a form at the end of every day. The form will contain questions regarding how much they are currently craving smoking and how much they smoked that day. Furthermore, participants will be split into two groups, one of which will act as a control, whilst the other will receive a set of smoking-related stimuli before filling out the form. Such stimuli will be given each day. This data can then be used to form correlations between higher-order associations and severity of smoking addiction. This will be further separated by the earlier assigned moderator variables. Thus, unique differences in data of correlation regarding how often one smokes or how long they have been smoking for can be determined, and further correlated to gender and age. Such correlations can be used by psychologists to determine which associations are optimal to focus on when designing smoking addiction management